So, breast cancer is the most common and deadliest type of cancer among female patients, and the survival rate of patients drops drastically from 99 to 31% once the cancer cells have metastasized. Previous studies showed that cisgingerol, a compound found in ginger, holds promise as a preventative treatment against metastasis by inhibiting factors such as cell migration and the expression and activity of metalloproteinases of MMP or MMPs. MMPs are enzymes that degrade the extracellular matrix that holds cells together, allowing tumor cells to escape a primary tumor, enter the bloodstream or lymphatic system, and spread throughout the body, therefore metastasizing. Therefore, we hypothesized in this study that treating MDA-MB231 breast cancer cells with a ginger extract containing cisgingerol would decrease cell migration and expression. Specifically the expression and activity of MMP2 and MMP9. To test this hypothesis, a ginger extract was prepared using ultrasound-assisted extraction, and then the cisgingerol content in the extract was quantified using UPLC. Then MTT assays were used to determine the sublethal doses of treatment in MDA MB231 cells. And after determining those doses, cell migration was evaluated using a scratch assay. MMP expression was evaluated using RT-qPCR, and MMP activity was evaluated using gel cymography, as you can see in the methods section right there. If we move on to figure number 1, the MMT assay results show that a 48-hour treatment significantly decreased cell viability at all doses. So we move on to analyzing a 24-hour treatment seen in part A of the figure, and that one only showed a significant decrease starting with doses of 5 micrograms per milliliter of cisgingerol. So from these, we chose 3.5 and 5 micrograms per milliliter doses as our doses in treatments for the remaining of the experiment. Now, when we analyze the cell migration in figure 2, the scratch assay showed that both of these doses cause a significant decrease in cell migration compared to the vehicle control, as seen in the images, as well as in the graph in part D of the figure. And the inhibitory effect was the same for both 24-hour treatment or 40-hour treatment. Therefore, 24-hour would be enough to inhibit the cell migration. However, when we move on to the RT-qPCR results in figure 3, there was no significant change in MMP gene expression, but when we look at the gel, the amplicons for MMP2 and MMP9, there were more than two amplicons in there, more than one amplicon, suggesting non-specific binding of the primer for these genes, and therefore this process seems to require a little more optimization or a new set of primers to be able to confirm the validity of these results. And in the case of thymogram in figure 4, no MMP activity was observed in the absence of these white bands against a dark background that is expected for MMP activity. There is the presence of a lot of proteins in all of the samples, but the activity of MMP was not seen. This could be because there was not enough MMP concentration in our samples. This wasn't completely unexpected, and it suggests that although costly, the only way to maybe perform a successful thymogram might be using a spin concentrator for proteins. In the end, these studies show promising results of cisgingerol as an inhibitor of cell migration in MDA-MB231 breast cancer, which could inhibit metastasis. However, no effects of MMP expression or activity was observed, and that since this is a contradictory results based on previous studies, this part of the experiment should be repeated, and the changes previously noted should be implemented to confirm whether this is a viable treatment. Further research could also be done by treating different types of cells with these ginger extract, other types of metastatic cells, be it other breast cancer cells or prostate or colorectal cancer cells.